Today, we're doing a deep dive into containment. It's a concept that's really vital, especially when you're working with biological processes, making absolutely sure that what needs to stay inside stays inside properly. So our goal today, unpack the exact steps, the practices used to stop harmful biological agents escaping. We're looking at particularly biotech stuff like fermentation and what comes after downstream processing. And guiding us through this honestly fascinating world of biosafety is technologist Dr. Rama Yusvana. He's a senior lecturer at the Faculty of Engineering Technology of Tun Hussein on University of Malaysia at UTHM. He's going to help us really get what containment means and crucially why it's just, well, non-negotiable.
Thanks for having me. It's a really fundamental topic for modern biotech, so I'm happy to talk about it.
Okay, so when I first heard containment in this context, I sort of pictured, just like really tight lid on a jar. But working with living organisms, powerful processes, I imagine it's way more complicated than that. What's the real challenge? And why is containment so essential for something like fermentation?
Yeah, you're right. It's definitely more than just a lid. At its heart, containment means all the measures, the practices you put in place to stop harmful biological agents getting out unintentionally. And the why? Well, there are several reasons. Protecting the workers, obviously, protecting public health from any potential, spread, and safeguarding the environment. But it's also about making sure the process itself is safe and sound, and that the final product, its quality and safety, are guaranteed. Think about it like a sealed bioreactor. It's not just holding the culture in, it's protecting everything outside from what's inside and protecting what's inside from anything getting in. Or a microbiological safety cabinet. When you work with cultures, it contains any aerosols. It's a direct barrier.
Right, so it's not just keeping the potentially bad stuff contained, but also keeping the product pure. That's a really important dual role. And I gather it's not just one single thing, right? There are different kinds of containment, almost like layers of defense. Can you walk us through those?
That's a great way to put it. Layers of defense. And yes, they're definitely designed to work together or provide redundancy. We usually talk about three main types: primary, secondary, and tertiary. And you're spot on. The strength isn't in just one barrier, it's in having multiple layers. If one fails, others are there, a safety net. So first up is primary containment. This is your immediate line of defense. Physical barriers stopping biohazards right at the source, like that sealed bioreactor vessel we mentioned. That's primary containment. It keeps the microorganisms locked in the system, keeps the product pure, prevents exposure. Biosafety cabinets too, another classic example, and they give you a controlled space for handling infectious stuff, protecting the person doing the work, from splashes or aerosols. The main goal here is keeping things sterile and minimizing exposure right where the action is. And it's crucial to remember these aren't just install and forget things. They need proper maintenance, regular checks to make sure they're actually working. Even a tiny leak could be a problem.
Okay, that makes sense. The immediate bubble around the hazard. What's the next layer out?
Right. Moving outwards, you get secondary containment. This focuses more on protecting the people, the personnel, and the procedures they follow in the lab space itself. This is where personal protective equipment, PPE, comes in big time. Gloves, lab coats, maybe face shields, respirators. These create a physical barrier between the person and any potential nasties. But the gear alone isn't enough. It's the procedures that make it work, like really strict aseptic technique, washing hands properly, decontaminating surfaces and tools after you use them, and really careful handling of waste. Following these protocols really helps control the environment and cut down risk, even if, say, a primary barrier had a momentary issue.
So the human element, the procedures become a key part of the containment itself.
Got it. And a third layer. The third is tertiary containment. Now we're talking about the facility level, the whole building design and its operating procedures, stopping anything hazardous getting beyond the lab walls. Think about things like specialized HVAC systems, heating, ventilation, air conditioning, but fitted with HEPA filters. High-efficiency particulate air filters. These trap really tiny particles, even biohazardous ones, keeping the air clean, filtering it before it gets recirculated or vented outside. Controlled access zones are also tertiary. Only authorized people can get in, reducing the chance of accidental exposure. And equipment like autoclaves, those high-pressure steam sterilizers, they're often used at this level to zap any microbes on equipment or waste before it leaves the facility.
Wow, okay. Primary, secondary, tertiary. It's a really thorough system. That breakdown really helps visualize it. So let's make it concrete. How do these layers actually play out inside, say, a big fermentation tank where things are actively growing and changing? How does it work in practice there?
Yeah, it's a very dynamic process, and containment has to be built right in. First, consider the design of the fermentation vessels themselves. They are almost always sealed systems, tightly sealed. This isn't just to stop spills, it's to prevent any microorganisms or the liquid media escaping. That containment minimizes risk, keeps the product pure, protects the environment and people. It's all happening inside that sealed box, essentially. Plus, they have safety features built in, like pressure relief valves. Fermentation can generate gas, right? So these valves handle excess pressure safely, stop the vessel potentially rupturing, and often you'll see secondary containment trays underneath just in case there's an accidental leak, an extra safety net.
Okay, so the vessel itself is like a mini fortress.
Pretty much. Then you have the use of bioreactors. These aren't just simple tanks, they're sophisticated closed systems. They maintain really precise conditions, temperature, pH, oxygen levels, all controlled to help the microbes grow well and make the product you want. And because they're closed, they naturally stop contamination getting in from outside and keep the biohazardous stuff inside. Crucially, they have strict sterilization protocols, like in situ steam sterilization. That means sterilizing the reactor with steam right where it sits before you start a batch and after you finish. These protocols are absolutely vital, keeps things sterile, stops batches contaminating each other, ensures the whole process is safe.
And what about actually putting the microbes in or taking samples out? That seems like a potential weak point.
Good point. That involves the handling of microorganisms. It demands extreme care, always using strict aseptic techniques. That means sterilizing tools, working in designated clean areas, following protocols to the letter when you're moving cultures around, and this is exactly where those biosafety cabinets we mentioned earlier are critical. They provide that enclosed, sterile workspace with air filters. The HEPA filters, again, protecting the worker from any aerosols and protecting the culture from contamination.
Right, it's clear every single step is designed to maintain that sealed integrity. Okay, so fermentation finishes, yeah. But the job's not done. You got your product mixed in with all sorts of other stuff. How does containment work when you move into downstream processing? Extracting and purifying, does the focus shift?
It does shift somewhat because the nature of the handling changes, but the core principles stay the same. The risks might be different, so the application adapts. Downstream is all about, first, getting your desired product out of that soupy fermentation broth, then purifying it, getting rid of impurities, and finally formulating it so it's stable and usable. Techniques here involve things like centrifugation, filtration, chromatography, drying. Each one needs careful containment.
So what kind of equipment helps with that?
Well, for containment equipment downstream, you see specially designed tools. Centrifuges, for example, often have sealed rotors. Why? Because spinning things really fast can create aerosols, tiny airborne particles. Sealed rotors keep those contained. It's a common hazard point otherwise. Filtration systems will have containment features built in too, making sure any particles, potentially biohazardous ones, are trapped within the filter unit, not escaping into the lab air. Even things like chromatography columns used for purification might have spill trays built around them. Just in case of a leak, it's contained locally.
And the cleaning, the decontamination must be intense too.
Absolutely. Rigorous decontamination procedures are key. You have to clean and sterilize equipment regularly. Prevents cross-contamination between different batches of products. This often involves, again, steam sterilization or using chemical disinfectants quite extensively. Disinfectants knock down the microbial load on surfaces, tools, autoclaving handles, waste, and some equipment. And waste disposal itself is a critical final step. You can't just toss biohazardous waste in the regular trash. It has to be segregated, put in special biohazard containers, and then treated, usually autoclaved or chemically disinfected before it can be disposed of safely. That ensures you meet regulations and don't contaminate the environment.
It really does sound like an unbroken chain of control from start to finish. Every length matters. So we've looked at specific steps, specific gear. What about the bigger picture, the overall strategies, the kind of design philosophies or rules that tie this all together?
Yeah, that's where you see how individual measures fit into a larger safety framework. We usually group these strategies into three main areas: engineering controls, administrative controls, and PPE. Engineering controls are all about the physical design, the infrastructure, how the facility itself is built and equipped. This includes things like using specific materials for floors and walls, making them sealed, easy to clean, preventing anything seeping through. We've mentioned HEPA filters and biosafety cabinets and ventilation systems. That's a core engineering control for air quality, and the ventilation systems themselves are often designed very cleverly to control airflow, sometimes creating negative pressure zones.
Negative pressure, how does that help?
It means the air pressure inside the lab or a specific room is slightly lower than the pressure outside it. So if there's a small leak, Air flows into the room, not out. It helps keep airborne contaminants contained within that area. It's a fundamental design principle in higher-level biocontainment labs.
Ah, clever. Okay, so that's engineering. What about administrative?
Administrative controls are the rules, the procedures, the how-to for people, the human side of the system. This involves having really detailed standard operating procedures, SOPs, written instructions for basically everything. How to run equipment safely, how to handle materials, how to clean up, how to dispose of waste. Consistency is key. Then there's comprehensive training and certification for everyone working there, making sure people have the skills and knowledge to follow the SOPs, understand the risks, and use the equipment correctly.
I imagine training for emergencies is a big part of that too. What happens if something does go wrong, despite all this?
Absolutely. Emergency response plans are a critical administrative control. What do you do if there's a spill, a power outage, a fire? Everyone needs to know the procedures, who to contact, how to evacuate safely, how to manage a containment breach. Practice drills are common.
Right? And the third part of the strategy.
Back to personal protective equipment, or PPE. It fits into the overall strategy as the last line of defense, right at the individual level. We've talked about gloves, coats, masks, shields. The key here, beyond just having it, is using it correctly. That means proper procedures for putting it on donning and taking it off doffing. You can contaminate yourself taking off dirty gloves if you're not careful. And just like the big equipment, PPE needs regular checking and maintenance. Is it damaged? Expired? Does it still fit properly? Making sure it's always functional.
It's just incredibly thorough. Layers upon layers of planning and control. It really paints a picture of how seriously this is taken, and given all that complexity, all that potential risk, it must be super tightly regulated, right? What kind of guidelines or standards are in place to make sure everyone follows these practices?
Oh, absolutely. The whole field operates within a strict framework of regulations and standards, both national and international. And these aren't just arbitrary rules, they're often built on decades of experience, sometimes, sadly, lessons learned from past incidents. They set the benchmarks for safety.
So what are some key examples?
Well, you have biosafety guidelines. Globally, the World Health Organization, the WHO, provides guidelines covering things like risk assessment, how labs should be designed, best practices for working safely. Then, nationally, you often have specific bodies. In the US, for example, the CDC Centers for Disease Control and Prevention defines biosafety levels, BSLs. They go from BSL-1, low risk, up to BSL-4 for the most dangerous pathogens. Each level has increasingly strict requirements for facilities, equipment, and procedures. And if you're working with, say, genetically modified organisms, there are often specific rules too. Like the NIH National Institutes of Health guidelines in the US for recombinant DNA research. They cover risk assessment, containment for GMOs, approvals needed.
Okay, so specific biosafety rules. What about broader industry standards?
Yes, there are also general industry standards that apply. Things like ISO standards, the International Organization for Standardization. ISO 9001 is about overall quality management systems. ISO 13485 is specific to medical devices, which can be relevant. They help ensure consistent, high-quality processes. For making medicines, pharmaceuticals, there are good manufacturing practices, or GMP. These are strict regulations enforced by agencies like the FDA in the US. GMP covers everything from raw materials to facilities to training, ensuring drugs are made safely and consistently. Very relevant for biotech products. And finally, you have worker safety regulations like OSHA, Occupational Safety and Health Administration in the US. They mandate safe working conditions, proper training, use of PPE, protecting the people doing the work.
Wow. Dr. Yusufana, that was an absolutely incredible journey into this mostly unseen world of containment. From the tiny scale of a sealed vessel right up to the whole building design, and all backed up by these global rules. It's really clear how vital and how, well, multi-layered this whole approach to safety is. You've really helped us grasp the sheer amount of thought and precision that goes into protecting the workers, the product, and, all of us.
Indeed. And if you think about the bigger picture, these principles of containment, they're fundamentally about managing risk, protecting life. It actually raises an interesting question, perhaps for you listening. Beyond these specialized labs and factories, where else in our everyday lives might we unknowingly be benefiting from, or maybe even applying, similar sophisticated ideas about containment?
That is a really fascinating question to leave us with. Thank you so much, technologist Dr. Rama Yusufana, for such a clear, insightful, and honestly quite eye-opening deep dive today. And for everyone listening, maybe next time you think about safety, you'll consider all those hidden systems, those invisible shields, working a way to keep things secure.